How synapses pass through neurons. Signals are received and passed through neurons, which have a main cell body with a nucleus and dendrites. And then they pass through the axon to the axon terminals, which are attached to the neighboring cell and its dendrites. And we call this the pre-synaptic cell and the post-synaptic cell. So obviously the synapse travels this way.
Each branch end of an axon transmits information to another cell, which is called synapse, shown in the drawing. Synapses are essential to neural function. Neurons are cells that are specialized to pass signals to individual target cells, and synapses are the means by which they do so. At a synapse, the plasma membrane of the signal passing neuron comes into a close opposition with the membrane of the target cell.
So when an action potential depolarizes the plasma membrane of the synaptic kernel, it opens voltage-gated calcium channels in the membrane, which triggers an influx of calcium. The elevated calcium concentration in the terminal causes synaptic vesicles to fuse with the presynaptic membrane. Then, the vesicles release neurotransmitter into the synaptic cleft. The neurotransmitter binds the receptor portion of the ligand-gated ion channels in the postsynaptic membrane, opening the channels. Here, both Na and K+ can diffuse through the channels. The neurotransmitter is released from the receptors and the channels close. Synaptic transmission ends when the neurotransmitter diffuses out of the synaptic cleft, is taken up by the synaptic terminal or by another cell, or is degraded by an enzyme.
Neurotransmitters are stored in the synapse in synaptic vesicles clustered beneath the membrane in the axon terminal located at the presynaptic side of the synapse. Neurotransmitters are released into and diffused across the synaptic cleft where they bind to specific receptors in the membrane on the postsynaptic side of the synapse. Most neurotransmitters are about the size of a single amino acid. However, some neurotransmitters may be the size of larger proteins or peptides. A released neurotransmitter is typically available in the synaptic cleft for a short time before it is metabolized by enzymes pulled back into the presynaptic neuron through reuptake or bound to a postsynaptic receptor.
Membrane potential is essentially the difference in charge between the outside and the inside. So commonly in neurons we'll see a membrane potential of between negative 60 millivolts to negative 80 millivolts.
Postsynaptic potentials. These graphs show changes in the membrane potential at a postsynaptic neuron's action hillock. These charts show the resting potential and action potential.
An action potential is generated as sodium flows inward across the membrane at one location. The depolarization of the action potential spreads to the neighboring region, reinitiating the action potential there. To the left of this region, the membrane is repolarizing as potassium flows outward. The depolarization-repolarization process is repeated in the next region of the membrane. Depolarization must pass a certain threshold of about negative 55 millivolts. Positive feedback will then propagate the signal and trigger the action potential. This threshold allows for an myelin sheaths are made of glial cells like oligodendroid or Schwann cells. They cover the axon and only expose small sections of axon to extracellular fluid, called the nodes of Ranvier. That was beautiful.
Can we try that again?